Hello, my name is Professor Hudson, and you are about to view narrated PowerPoint lectures for Chapter 9, which deals with mitosis and cancer. For this segment, we will talk about how is the DNA in eukaryotic chromosomes organized. Let us begin. Eukaryotic cell division is more complex than prokaryotic cell division due to the complex way in which eukaryotic cells organize their genetic material. In eukaryotic cells, the eukaryotic chromosomes are separated from the cytoplasm by a membrane-bound nucleus. Eukaryotic cells always have multiple chromosomes, and eukaryotic chromosomes are longer and have more DNA than prokaryotic chromosomes. Let's take a closer look at the organization of a eukaryotic chromosome. The eukaryotic chromosome consists of linear DNA, double helix bound to proteins. Each human chromosome contains a single DNA double helix about 50 million to 250 million nucleotides long. Most of the time, the DNA in each chromosome is wound around proteins called histones. These DNA histone spools are further folded into coils. And then another layer of folding occurs at the coil strand where it folds into loops. So now the chromosome is 1,000 times shorter than when it was extended. During cell division, more proteins fold up the DNA in histones until it is 10 times shorter than in its resting state. The chromosomes contain segments of hereditary information called genes. So in cells, most of the time, the DNA is wound around proteins, and it further compacts around proteins when the cell is about to divide. Genes are segments of the DNA of a chromosome. Genes are sequences of DNA from hundreds to thousands of nucleotides long. Each gene occupies a specific place or locus on the chromosome. In addition to genes, every chromosome has specialized regions that are crucial to its structure and function. All chromosomes will have telomeres, centromeres, and the telomeres consist of repeated nucleotide sequences which are essential for chromosome stability. One theory on why people age is that as people grow older, the telomeres shorten and the DNA is less protected. The DNA is more susceptible to degradation and mutation. As these mutations and errors and degradation occur, the cell does not function as well as it should. The second specialized region of the chromosome is the centromere, which has two principal functions. It temporarily holds the two daughter DNA double helices together after DNA has replicated. And it is the attachment site for microtubules that move the chromosomes during cell division. Let's take a look at a karyotype. A karyotype is a pictorial display of the chromosomes found in a cell. Karyotypes are often done when a researcher wants to analyze the number and shape of chromosomes. The number and shape of chromosomes can sometimes identify certain abnormalities that lead to syndromes, such as Down syndrome. Humans have 46 chromosomes. When a cell contains the full set of chromosomes that species is supposed to have, we refer to that cell as diploid. All of the cells in your body are diploid except for the sperm and egg, which are haploid. Haploid cells have only half of the full set of chromosomes. Why do you think the sperm and egg are haploid? Yes, if you said that sperm and eggs are haploid so that the chromosome number is maintained when the sperm and egg get together during fertilization, you are correct. If the sperm and egg were not haploid, upon fertilization, the number of chromosomes would double each time a sperm and egg get together. One of the things that can be noted from the karyotype is that chromosomes usually occur in pairs, which contain the same genetic information. Both members of each pair are the same length, shape, and have the same staining pattern. These similarities occur because each chromosome in a pair carries the same genes arranged in the same order. Chromosomes that contain the same genes are called homologous chromosomes or homologs. A typical human cell has 23 pairs of chromosomes for a total of 46 chromosomes. Half of these chromosomes came from mom and half came from dad. 22 of the 23 pairs are called autosomes, and then the 23rd pair is called the sex chromosomes. If you are male, your sex chromosomes will read X and Y as shown in this figure. If you are female, your chromosomes for the sex chromosome should be XX. If you have something called Down syndrome, instead of 46 chromosomes, you would have 47 chromosomes. And this karyotype would show an extra chromosome for the chromosome pair 21. This slide shows a pair of homologous chromosomes. Note they contain the same genes in the same location. One of the pair members is from mom and the other member of this pair is from dad. The homologous chromosomes like to be near each other because during meiosis they will exchange DNA in order to mix up the gene pool. We will talk about meiosis in another module. Now, prior to cell division, DNA must be replicated so the daughter cells can receive the same number of chromosomes as the parent cell. During DNA replication, a single chromosome is copied, with the copies remaining attached to the parent chromosome at the centromere. At the end of DNA replication, the duplicated chromosome consists of two identical DNA double helices called sister chromatids. So a sister chromatid or chromatids are used to refer to the fact that the chromosome has copied itself. In this figure, we have a chromosome. In part A, we have an unduplicated chromosome. In part B, we have a duplicated chromosome. Note that the duplicated chromosome consists of two chromatids. The whole thing is called one chromosome, and then each half is called a chromatid. The sister chromatids are attached at the centromere, and they are genetically identical. Now that we have talked about the organization of the chromosome, let's talk about cell division with mitosis. The goal of mitotic cell division is to produce two genetically identical cells with the same number of chromosomes as the parent cell. Let's continue and talk a little bit about what happens during the eukaryotic cell divisional cycle and how mitotic cell division produces genetically identical daughter cells. Let's talk about interphase, which is the first part of the cell cycle for cell division in eukaryotic cells. As mentioned previously, interphase consists of G1, S, and G2. Interphase is a time for acquisition of nutrients, growth, and chromosome duplication. During interphase, the DNA and associated proteins are in a tangled mass of thin threads called chromatin. These figures show animal cells in interphase. Notice the purple region, which is the nucleus with the chromatin. You cannot see individual chromosomes at this point. During interphase, a cell grows in size, replicates its DNA, and often differentiates. G1 is the time for acquisition of nutrients and growth to proper size. Cells will grow in size. They can specialize and differentiate depending on the cell type, which means entering a fifth stage called G0, or they can decide whether to divide or not. Once again, if the cell decides not to divide, it can go into the resting stage G0. Then there's the synthesis phase, which is characterized by DNA replication. And then there's another growth phase called G2, which includes completion of cell growth and preparation for division of the cell into daughter cells. So once again, interphase consists of G1, S, and G2. G1, the cell grows and differentiates if necessary. S, the DNA is replicated. In G2, the cell grows and prepares for mitosis and cytokinesis. This figure shows the entire cell cycle from start to finish. We will talk about each phase in more detail. Once again, the interphase is G1, S, and G2. This is where the cell gets ready for the division and duplicates DNA. Mitosis follows G2, and this is where the cell will actually separate the duplicated chromosomes. And cytokinesis also occurs to try to divide the cell in half. The cell cycle is carefully controlled throughout the life of an organism. Without enough cell divisions at the right time and in the right organs, development falters or body parts fail to replace worn out or damaged cells. Or if this cell cycle process does not occur as it's intended, too many cell divisions can occur, which can lead to cancer. Let us review. Please write down the correct answer for this question using the letter only, as you will need it later. Question 3. The DNA is replicated during which stage of the cell cycle? Choose the correct answer. This is question 3. Question 4. Chromosomes are composed of, choose the correct answer. Question 5. The region where sister chromatids are attached to one another is called the, choose the correct answer. Now let's take a look at what occurs during the eukaryotic cell cycle. Mitotic cell division consists of nuclear division and cytoplasmic division. Mitotic cell division is the division of one parental cell into two daughter cells. Mitosis is the division of the nucleus, and cytokinesis is the division of the cytoplasm and cell membrane. This slide shows an overview of mitosis, focusing on what happens to the chromosomes during the process. Note that early in interphase we have a single cell with 4 chromosomes. The 2 chromosomes from mom are in red and the 2 chromosomes from dad are in blue. During S phase of interphase, this single cell will reproduce or replicate the DNA. Notice we have 4 chromosomes still, but each chromosome has 2 chromatids. So I have 4 chromosomes and 8 chromatids. Then mitosis occurs where this single cell will divide into 2 cells. And each one of these chromosomes will separate at the centromere, leaving each of the daughter cells with 4 chromosomes. The initial cell that we started with had 4 chromosomes, and the daughter cell has 4 chromosomes. 2n equal 4, which means the diploid for this cell is 4. Let us pause here In the next segment, we will go through the stages of mitosis in more detail.